Today I'm going to be presenting work that I've been doing with my niece Picard, pictured here on the screen. And we've been looking at using sedimentary eDNA to detect the presence and impact of European perch in New Zealand lakes. This project is part of the Lakes 380 program, a nationwide project looking at lakes health in the past, the present and the future. So this has also been a team effort to put this together. So the lake that we're interested in today is Lake Pounui. And Lake Pounui is a lowland lake in the south of the North Island in the Wairarapa region. And it's quite unique in New Zealand, as it's one of the few lowland lakes that has its majority of its catchment in native vegetation. Despite this, in the past few decades, it's been experiencing a decline in water quality and an increase in cyanobacterial blooms. And this work that I'm going to be talking to you about today is off the back of some mesocosm work that was done actually in Karori Reservoir in the Wellington region that was looking at the relationship between European perch and cyanobacterial blooms, and found that with an introduction of perch to an area of the lake, there was an increase in blooms. And so we wanted to understand whether this driver and the introduction of European perch in Lake Pounui was having a similar impact. So to introduce you to Lake Pounui and its disturbance history, we start off around the 1400s during the period of Māori settlement. And then during this time, there was some catchment clearance, but the main catchment clearance really happened in 1814 with European settlement. And it was at this stage that a lot of burning happened in the catchment. And then there was a regeneration of the native forest, and then intensification of land use happened in the 1950s, where chunks of the catchment were cleared, and there was also aerial top dressing as there was conversion to pasture land. At the same time that these catchment stresses were happening, we also saw an increase in air temperature, and this correlated with increasing annual mean temperatures of the lake, as at around 14 degrees Celsius in the 1980s, and it's increased to about 16 degrees Celsius in 2008. At the same time, we have a range of in-lake stresses, and I'm highlighting today the introduction of non-native fish. So from around 1930 to 1950, we have a record actually of rainbow trout being stocked into this lake. And then fish surveys started being taken from about the 1970s, and we see both brown trout and European perch popping up in these fish surveys. And so we're really wanting to link these stresses to the lake responses we see, which, as I mentioned before, is this transition from a mesotrophic to a more eutrophic lake in present days, with observed cyanobacterial blooms, and cyanobacterial blooms occurring most summers nowadays. And we're really interested in trying to elucidate the impact of European perch within this whole catchment story. And so to do this, we use paleolimnology, and this is essentially, we go out into the middle of the lake, we take a sediment core, and we use the sediment core as an archive, a mud archive of time and environmental history of this lake. We take it back to the lab, subsample down core at certain time periods, and then we extract eDNA from this mud. And we really had two approaches. We firstly wanted to identify the arrival and presence of European perch in our lake, and we used a targeted species approach, namely droplet digital PCR. And then once we'd done this, we wanted to determine the biological impact of European perch. And to do this, we looked at aquatic communities using a metabarcoding, so a community-based approach. And I'm firstly going to talk through our results of our droplet digital PCR approach. So using sedimentary eDNA to detect the arrival and presence of perch, as you can see here, this figure shows us a timeline of from present day sediment at the very top of the graph all the way down to pre-human. So this is more than 1,300 years ago. And I've separated this graph into three main time eras. So you have post-European settlement So you have post-European settlement at the top, a period where there's evidence of Moody settlement, and then pre-human, prior to any human arrival around the catchment. And the dots highlight where we have detection of perch DNA in our environmental DNA sample. And as you can see, all of these positive detections are occurring post-European settlement and really early in European settlement. So this was awesome because we managed to detect European perch in our sediment, and also it gave us some evidence around when that perch might have been first introduced into this area. So prior to this DNA work, we knew that perch were observed in fish surveys in 1977, but this data indicates that they've actually been in the lake much earlier than this, up to 100 years earlier. However, there are a couple of method considerations when we're looking at this data. Firstly, the modelling of our age data and linking it to sediment depth has some uncertainty. So although we've identified 1850s as this point of first detection, this can be a much broader range and can also increase due to bioturbation. So that's something to keep in mind. Secondly, in this period post the first detection of perch, we see some samples where we're actually not getting any perch detection. And so this patchy detection is not what we'd assume, because we'd assume once the perch are in the lake, we'd have a population that's maintained pretty consistently throughout time. And this patchy detection we think is more likely due to heterogeneity in DNA deposition and preservation. So it's more of a method limitation, but it's important to take into account when we're looking at these results. And finally, that this approach can be species dependent. So I mentioned earlier that both brown and rainbow trout have been found in the fish survey records in this lake. However, despite this, we had a look for them using droplet digital PCR, and we didn't get any positive detections. And we think this is largely due to biomass. So neither of these fish have been found in very high biomass in the lake, and this could hinder our detection, but is also important to consider when we're looking at low abundance species. So now that we've identified that perch arrived early in this European period, we wanted to understand their biological impact in the environment, and we wanted to specifically look at their impact on aquatic communities. And we did this using metabarcoding. And we chose three main groupings. So we had bacteria, a range of micro and macro eukaryotes, so algae, fungi, invertebrates, worms, and also macrophytes. And the hypothesis behind the impact of European perch on this lake is that European perch eat zooplankton and other invertebrates that are responsible for grazing on phytoplankton. And so this increase in predation releases grazing pressure on the phytoplankton and cyanobacteria and allows blooms and phytoplankton dominance in the lake. And so we wanted to understand if we could see these changes occurring after the introduction of perch to our lake. So this graph shows these three different groupings. For eukaryotes, we actually had two gene approaches. We used both ATNS and CO1. And this is all community data, but we're showing it as a principal response curve score. So this is essentially the change in community composition over time. And we've also applied a GAMS analysis over the top of this. So this is the generalized additive mixed modeling approach, which identifies periods of significant change in our communities. And these are highlighted by this blue or red lines in each of our communities. And so if we look at these periods of significant change and correlate them to where we see our trout first being introduced into our catchment, we can see that they line up pretty well following the introduction of perch into this ecosystem. And so this kind of highlights that perch may have played a role in initiating some of this change and perhaps causing some of the continued change in biological communities over time. However, we don't want to look at this data in isolation, and I'll take you back to the kind of environmental disturbance history of this lake. And throughout time, there have been low levels of catchment used, especially during the Moody settlement period. And then this increase in land use change during the European settlement period. And this is just important to consider that while we're looking at perch impacts, some of these changes may be related. These, though not extensive, still present land use changes. And especially when we hit the 1950s, we see the addition of aerial top dressing and pastoralization in the land. And so these kind of more present post-1950s impacts may be a result of this land use change, and in addition, even temperature increases in the lake. And so we think in concert with the perch introduced changes, we're also seeing these communities respond to a wide variety of catchment disturbances. I also wanted to highlight that within this metabarcoding data, we also found some other information about invasive species, specifically macrophytes. So in present macrophyte surveys in Lake Pounui, we have identified a range of exotic species that exist in the lake. And using this kind of same technique, we can now identify when they were first introduced or they first came to the lake, and start thinking about the role they may have had to play in the changing environment in the lake. And these three invasive species are known to outcompete native macrophytes, and that's one of their main detrimental impacts in an ecosystem. So just to kind of wrap up with some conclusions, firstly, we showed that we can use the sedimentary eDNA approach in historical sediments to identify the arrival of the non-native European perch to Lake Pounui. And interestingly, it's around 100 years before we observed it in fish surveys, giving us new evidence for how long perch may have been impacting this lake ecosystem. Following on from this, we can actually correlate this perch arrival with shifts in different aquatic communities. And one of the maintained hypotheses is that perch predate on the zooplankton, leading to phytoplankton dominance, and could be one of the drivers of these cyanobacterial blooms in the present-day lake environment. But we have to consider this introduction of perch alongside other environmental stresses such as land use change and also climate, because it was more likely that a holistic approach and also additive impacts are being seen within the community shifts in the aquatic environment. So sedimentary core eDNA can be used to identify the presence or absence of a non-native species in the sediment record, with some considerations such as biomass, the type of species, and also lake type. And then we can use this within a greater ecological context to infer ecological impact, knowing different aspects of the catchment environmental disturbance history. So I'd just like to thank you very much for listening today. And I have a lot of acknowledgements, Victoria University, the local iwi of Lake Pounui landowners, the Greater Regional, Wellington Regional Council, and Lake Seriety in general for allowing me to do this work. Yeah, and I'll be happy to take any questions.
Awesome. So I think we should have Georgia on Zoom, right? So if anybody's got any questions. Do we have you on there?
Yes, I think so.
Oh, excellent. All right, now we hear you.
I don't know if you can see me though.
No, but at least we can hear you.
Okay.
Awesome. Thank you for a really interesting talk. I was just curious, you highlighted with the macrophytes that you found some invasive species and were able to date when they came in. I'm wondering if you noticed anything else, any other interesting species introductions or changes driving some of those other community shifts that you found?
So I wasn't specifically focused on the other than perch, actually. I just brought the invasive macrophytes into the picture because I thought everyone might be interested. So I haven't done a deep dive into all the other invasive species, but definitely now we can kind of put a date to some of these timings. We can potentially look a bit more intricately at some of those changes. But right now we're kind of focused at this high level total community shifts that we can see. Yeah, there are some limitations around taxonomically identifying some of the species within the bigger community groups and stuff like that. But yeah, it would be awesome if we could take a better deep dive, especially in the macrophyte community area. Thank you.